Good evening. I'm Dan Gill with Allergan, and it's my pleasure to welcome you all this evening. First off, I have some business items to attend to. First off, just to remind everybody, please turn off or silence your cell phones, mobile devices. Secondly, did everyone receive your 3D glasses when we came in? I think if you haven't, Brian up there, raise your hand if you haven't, and Brian can bring them to you. So keep them up, and he'll come around and bring them to you. So our goal in the Allergan lecture series is to have speakers who help us understand how basic biological research helps us better understand our lives in the world we live in. And I think tonight's speaker will certainly, in a very enlivening way, help us understand a lot about the visual system. My job, though, before we get to the speaker, is to introduce a very good friend and collaborator in sponsoring this series, and that's Al Bennett, who is the Hannah and Francisco J. Ayala Dean of the School of Biological Sciences, and he's been in that role since August of 2007. He's been a professor here at the university since 1974, and prior to that, prior to being the dean, he served as chair of the school's Department of Ecology and Evolutionary Biology for 8 years and as chair of the Department of Development and Cell Biology for 3 years. Dr. Bennett's own research focuses on evolutionary and comparative physiology of animals with special emphasis on how they adapt to diverse environmental factors. He's a fellow of the American Academy of Arts and Sciences and also of the American Association for the Advancement of Science. And so, without further ado, I'd like to welcome Dean Bennett, who will introduce our speaker.
Thanks very much, Dan. It's a pleasure to introduce Margaret Livingstone. She's a professor of neurobiology at the Harvard Medical School. She received her, she's from the East Coast, so we're privileged to have her out to see the other ocean. She received her Bachelor of Science from MIT and then got her Ph.D. in neurobiology at Harvard, and she has been at Harvard in the medical school for over 30 years now. One of her first research breakthroughs occurred when she and a collaborator discovered a new subdivision of the visual cortex that involved the processing of color. So this, her interest in color, vision, etc., goes way, way back in her academic studies. This area, this work has had profound impacts on many fields of study relating to perception and physiology. She has been involved in the study of numerous other aspects of neurophysiology, including visual processing in subjects with dyslexia, which impact the field of studying learning disabilities. She studied face recognition and processing in monkeys. And she's used MRI technology to localize the function of neurons within the temporal lobe. This has significant implications for the study of autism. Lastly, and what she'll be talking about today, is that Margaret's explored the ways in which vision science can understand and inform the world of visual art. She's written a popular book, Vision and Art, which has sold more than 30,000 copies. And this work has brought her acclaim in the art world as a scientist who can communicate with people in both as both artists themselves and art historians. Here at UCI, one of our strengths lies in having fields which cut across our entire campus for interdisciplinary work. And Margaret's work speaks to many of the different schools on campus. She's known quite well within our department in biological sciences, in neurobiology and behavior, but also our colleagues across campus in cognitive sciences, in the School of Social Sciences, and in the Claire Trevor School of Arts know of her also. And many of us have been looking forward to hearing her talk today. And so I'm very pleased to present to you, Dr. Margaret Livingstone.
Thank you, and I'm pleased to be here. I am a visual neurophysiologist, which means that I try to figure out how we see by recording from neurons in the visual system. But I acknowledge that artists also try to figure out how we see, and some works of art are important or great because they reveal important things about how we see. Now, I know some works of art are important because they reveal important things about human culture or human psychology, but I'm not going to talk about those. I'm going to talk about works of art that reveal important things about low-level vision. And the first thing I'm going to talk about is the use of line drawings. So artists have always used lines to represent contours, but there are no lines out there in the world. There are contours and discontinuities, and yet we see lines as contours and artists represent contours as lines. Why does this work so well? It has to do with the way your visual system processes visual information, and the first and most fundamental stage in visual information processing is center-surround antagonism. So Steve Kufler discovered that if you shine a spot of light on a retina, you get a barrage of signals from a retinal ganglion cells when the light is turned on. But if you make the spot larger, that is, you put more light on the retina, paradoxically you get a worse response. And Steve deduced that this means that retinal ganglion cells are excited by light in one part of the visual field, but they're inhibited by light in the immediate surround. So if he shined an annulus of light on the retina, he got a suppression of firing. This is the first and most fundamental stage in visual information processing, and in case you don't believe me that your visual system starts with center surround, do you see the little gray dots everywhere where you're not looking? Don't worry about why it's where you're not looking, that's postgraduate. The reason you see the little gray spots is because center surround cells at the intersection have more white in their inhibitory surrounds than center surround cells at the crosspieces, so they're less active. There's an extra synapse in calculating the surround antagonism. You have to go from excitation through an inhibitory neuron to inhibition, so that there's a little temporal delay, which gives you this sparkly quality in some of these versions. If you take anything home tonight, I hope you will take home the idea that vision is information processing, not image transmission. It doesn't do you any good to transmit a high-resolution image to your visual cortex because there is nobody up there to look at an image. All you've got up there are neurons, and all neurons can do is fire or not fire. So vision is all about translating the pattern of light on your retina into a pattern of firing or not firing of nerve cells at different stages in your visual system, and it's about extracting information from the environment into this pattern of firing of neurons. And neurons are stupid. So vision, the computations have to be simple. All they can do is fire or not fire. Local, they're little bitty things. They're single cells. They can communicate with nearby neurons, but that's about it. And it's often opponent because nearby neurons often inhibit each other. And I said that, right? It's simple. All they can do is excite and inhibit each other. It happens between nearby neurons, and they often inhibit each other. So why do I say that center surround is information processing? Well, if you had an image like this that was white on one side and dark on the other, center surround cells over here are going to be inhibited. Center surround cells over here are not going to be activated. It's only center surround cells at the discontinuity where there's an imbalance between the center and the surround, that you get activity. So center surround cells turn a contour into a line drawing. And that's information, because in an image, where the edges are is where the information is. And if you were going to save this as a file on your computer, as a TIFF file, where you save the value of every pixel in the image, it would be a huge file, right? Megabytes. But if you save it as a JPEG, which compresses the image, JPEG only tells you where the edges are and what the sign of the contrast is across the edge, so it saves information. And this is just to remind me how efficient a line drawing can be at conveying information. But it also allows me to point out, what color are these lines? Come on, this is easy. Black. What color is this screen? White. But you know that the amount of light coming to your eye from that black has to be the same as the amount of light coming to your eye from this white, right? That projector can't project blackness, it can only project light. So your visual system doesn't care how much light there is at any point in the visual scene, it only cares about contrast. And your visual system and your brain in general care about all kinds of contrast. So you care about contrast in contrast. These checks are the same as these, but these look higher contrast because they're surrounded by lower contrast. We have contrast in local contrast in orientation, so these lines are vertical, but they look away from vertical because they're surrounded by not vertical. That dot is moving vertically, but it doesn't look like it is because of the contrast in motion. All these dots are identical grays, but this looks less yellow or more blue, and this looks more yellow or less blue because of color contrast. So color shows contrast both locally and spatially, like luminance. And so here's a little illusion based on that. I want you to look just for a few seconds at that black dot and remember that what you're looking at is white letters, and now you're going to get a yellow after image from those white letters because of the surround opponency of color. Great artists, of course, know that. Monet painted this haystack with a blue shadow because skylight is bluer than sunlight, and he even put a little mock band in yellow around the edges. We also compute depth and spatial organization, and we start computing this locally. So you can spend lots of money on a computer program that will do what's called ray tracing, where it will make sure that all the light sources in your image are perfect and that things bounce off in physically plausible ways. Here's something that was made with a ray tracing program. But it is completely unnecessary. You don't need to buy a ray tracing program because the computations about depth start in a part of the visual system where the computations are local. That is, in the visual system, in the first cortical visual area, receptive fields march across the visual field in a systematic way, and they're smaller in the fovea and bigger as you get farther out. So space is represented in cortex. So nearby cells care about the same part of cortex. Furthermore, one part of cortex, which deals with one part of the visual field, is far away from the parts of the cortex that deal with other parts of the visual field. So depth computations are local. Now, what do I mean about depth computations? So the world looks vividly three-dimensional, right? I mean, I know it is, right? But the way you see it as vividly three-dimensional is with two flat retinal images. You start with two flat retinal images, and from those two flat retinal images, you get all this information about distance and depth. It's not like sonar. You don't ping anything. And so your visual system has to compute distance and depth, and it does it with a number of cues. It uses relative motion, so when you move, things that are near move more than things that are farther away. Shading, we know about that. Perspective, occlusion, things that are in front of others, and stereopsis, which I'll talk about at the end. But because these computations start in early visual cortex, even though depth is a global perceptor, globally physically, the calculations are local, and artists have ignored the laws of physics for thousands of years without anyone caring. you can afford to ignore the laws of physics. You don't need to go around checking them. They're going to be obeyed no matter what you do. So you can have shadows that go in completely different directions. You can have a vase of flowers in front of a window with the shadow going towards the window. No one cares. This is Canaletto's Piazza San Marco, which is at Harvard, at a little art museum in Harvard. And the shadows are going in this direction, suggesting that the sun's over there. But in this part of the painting, the shadows are going that way. When I pointed this out, the curator at the Fogg said, "Oh, that's because the sun is reflecting off this building and casting shadows in this direction." So I went to the Piazza San Marco in Venice. This building is just covered with windows. It does not reflect light and make the shadow. The shadows in the Piazza San Marco all go in the same direction. No one cares. In this painting by Edward Hopper, here's a window casting light on this wall, but this thing doesn't cast any shadows. No one cares. Here's a manger that forms a Penrose figure in that you're seeing it from above over here and from below over here. I suppose that there's no real reason why halos should even think about obeying the laws of physics, but in this painting, whatever it is they're doing isn't even internally consistent, and I think Matisse was playing with the idea that even occlusion cues don't have to obey the laws of physics. If you can find a painting that has a mirror in it, I guarantee you it's not obeying the laws of physics, because if this lady were looking at herself, you wouldn't see her face in the mirror, right? Vermeer paints this nice shiny brass bowl, and it looks shiny because the bowl is reflecting the tablecloth, but if you look a little more closely, you'll see that what the bowl is reflecting isn't the tablecloth underneath it. So another thing that artists figured out a long time before us neuroscientists is that color and luminance do different things, and I'll tell you what luminance is in a second. Picasso said, "Colors are only symbolic. Reality is to be found in luminance alone." So we have 3 cone types in our retinas: short, middle, and long. George Sperling, close your ears. Some people think of them as blue, green, and red. We see color by subtracting the activity in different pairs of these cones. We see luminance just by adding them all together. It's like black and white photography. So in this image, this is the color version, this is the luminance version. For those of you who are in the art field, I mean value, and I'll try to remember to say that sometimes. Most color contrast borders also have a luminance contrast, but you can have color contrast borders that have no luminance contrast, and special things happen when you do that. And I think the Impressionists discovered this several times over and over. So this is Impression Sunrise by Monet. It's the painting after which the Impressionist movement got its name. And in the original, you need to go to Paris, I'm sorry, the sun absolutely shimmers. It has this kind of hot, cold, vibrating quality. And it seems really bright. But I once got a small grant from Harvard to go to Paris and measure the luminance of the sun in Impression Sunrise. And it turns out that despite the fact that it looks really bright, it's actually exactly the same luminance as the background. And I think that's why it has this peculiar quality, because the human visual system and the primate visual system in general starts in the retina, goes on up to the thalamus, primary visual cortex, and then it splits into these 2 major processing streams. And we know about what these streams do, largely from neurology, because of what happens to people when they get lesions in different parts of their visual system. So people who get lesions in this ventral stream lose the ability to recognize objects. They get agnosias. They can copy something, they can see it, but they can't tell you what it is. So we call this the what system, because it seems to be largely responsible for object recognition, including faces and color perception. This part of our visual system is a primate add-on to the basic mammalian visual system, which is represented by this dorsal stream, which carries information about motion, depth, spatial organization, and figure-ground segregation. So here are some drawings made by 4 patients who had lesions in their wear system. Some people call it the house system because it seems to be responsible for how you interact with the environment. Or wear, doesn't matter. These patients were asked to draw a bicycle, a cross, and a clock. So you can see that these patients got all the bits right, but their spatial organization is messed up, kind of like cubist drawings. I'm not trying to say that Picasso had a parietal lobe lesion, but rather that what something is and where it is are 2 quite different things. Now, for the purpose of this talk, I want to talk about the fact that this part of our visual system, like other mammals, is essentially colorblind. So if you have a red spot on a green background and you take a black and white photograph of it, or you look at it with your wear system, you're going to get a gray spot on a gray background. And you can have different brightnesses of red that will give you different relative grays. And you could imagine having a red that gave you the same gray. That is, it was invisible to your wear system. That's what I'm going to call equal luminance. And very peculiar things happen at equal luminance because there's a whole part of your visual system that can't see it. So none of those were equal luminant, but for me, this one is close to equal luminant, and so's that one. They're kind of shimmery. They have this little weird quality. So I am going to try to convince you that your ability to see depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. So this thing looks 3-dimensional from relative motion, right? Of course, it's flat. I know it's flat. And it looks 3-dimensional whether the red is brighter than the background or darker than the background. But I can creep through equal luminance and make this thing look flat and squishy and then make it look 3-dimensional again. Let's go back through flat and squishy. I like this projector now. So now we go through equal luminance and it looks flat and squishy, and now it looks 3-dimensional again. And don't worry if it switched direction, that's postgraduate. So I hope you can see that this image has a lot of depth from perspective and depth from shading in it, but if you close one eye and look through the red lens of your glasses, I hope this thing will look more 3-dimensional because the red lens blocks the blue light. Okay, now, more 3-dimensional when you look through the red lens. Now, put the glasses down. Here's another version. Again, look at it and see if you can see that it's got a lot of depth cues, but it's kind of even hard to figure out what's going on, where's the building, what's going on in the background. And again, close one eye and see if you don't get a lot better sensation of depth, and you can even figure out what's going on. The whole image makes sense. You do better figure ground segregation. Okay, glasses down. Monet painted a whole bunch of versions of this cathedral in Rouen, and they're all about light and depth from shading because it's just a beige stone building. And you can see in this version, the luminance contrast shows the depth in the doorway and the rose window, but some of them, like this one that's at the MFA in Boston, look flat and shimmery. And this is at sunset, the building faces west, and there is no luminance contrast. And I think Monet managed to use colors that were very close in luminance to give this flat, shimmery quality to this building with the sun pouring into the doorway. And this is part of Lichtenstein's series called the Rouen Cathedral, and I bet it doesn't look at all like the Rouen Cathedral until, again, you close one eye and look through the red lens, and there it is. I think he was playing with that. Andy Warhol was playing with it, with the same concept that you don't get depth at equal luminance in his Brooklyn Bridge, so you can probably figure out that this is the Brooklyn Bridge and what it shows, but in the top half of this same image, I hope you see a lot more depth from perspective than you do in the bottom version. So I hope I've convinced you that your ability to see depth from perspective, depth from shading, and depth from relative motion are all carried by a colorblind part of your visual system. The corollary is that it doesn't matter what the color is as long as the luminance is appropriate to get depth signals. And I think, again, artists figured this out. This is André Derain's portrait of Matisse. The shadows on his face are very strange colors, but if you knew Matisse, you would recognize him. Because although the shadows are weird colors, the luminance is exactly right to give you the shape of his face. And in Matisse's Lady in a Hat, the same thing. I think he's doing what Picasso said. He's using colors symbolically, but the reality, the shape of her face, is there in the luminance alone. So I hope I've convinced you that your ability to see depth, spatial organization, and figure ground are carried by a colorblind part of your visual system. Now I'm going to try to convince you that your ability to see motion is colorblind. And again, I'm going to show you a little demo. So this demo shows some stripes. So I want you to look over here, okay? And whether the green is brighter than the red, when the green is brighter than the red, I hope you see that these two sets of stripes are moving at the same speed. Don't cheat and look at the center. Just appreciate the relative motion. And when the green is darker than the red, I hope they seem to be moving at the same speed. But as I go through equal luminance, and you're not cheating by looking at the center, I hope you experience the stripes on the left, which are close to equal luminant, as moving slower than the stripes on the right. If your ability to see motion is colorblind, your ability to see that something is not moving is colorblind. And I think Monet achieved a sense of motion in many of his paintings by using colors that were close in value or luminance. So the poppies are about the same luminance as the grass, so you can see them with your what system, but your where system can't see them, so they can seem to move. Monet's water, I think it moves because he uses splotches of equal value colors all over the place. Now, the art historians all say that Monet got motion in his water, but Monet didn't write about it. We don't know this, but I know that Pete Mondrian knew that he got motion in this painting because the title is Broadway Boogie Woogie. The original's at the moment in New York. It's about this big across, and it jumps around so much it's kind of nauseating. And I think it jumps around because the yellow checks are highly visible to your color selective what system, but your where system can't see them, so their position is indeterminate. Advertisers do this all the time. You open Wired magazine, you will find an ad or a headline that has this unpleasant, jittery quality. You can do it whenever you have pairs of colors that are equal luminant. And I think advertisers do this because the jumpy, jittery quality is attention-getting, and, as I will talk about at the end, it's hard to read. Why did I put this up? Oh, so I hope I've convinced you that your ability to see motion, depth, spatial organization, and figure ground segregation are colorblind and computed at least at a part of the visual cortex where receptive fields are small. Now I want to try to convince you that your ability to see color is coarse. You do not have to color inside the lines. And of course artists know this. Watercolors, pastels are characterized by a line with a sloppily applied color that comes somewhere near the line, and it works just fine. And that's part of the artistic interest. Here's an illusion based on this. I hope you see the donut as yellower than the whole or the background. Come on, you have to agree with me. Okay. It's not. I made this. It is a yellow line inside a purple line. The donut is exactly the same white as the whole and the background. It's just a yellow. Because your color acuity is so low, your visual system lets the color spread till it hits a border because it isn't sure where it is. So now I'm going to use after images to generate some blurry colors and let you watch them spread till they hit a border. What I want you to do is look right at that intersection of those three shapes, and then I'm going to show you an achromatic gray, and you're going to maybe see an after image. Now I want you to do it again. Look at the intersection and evaluate the after image in this achromatic. And some of you are saying, well, it was better the second time because you did it twice, right? Look at the intersection, evaluate the after image, look at the intersection, evaluate the after image. It's better when it's constrained. Now, look at the intersection and see if you can get the color to fill a different shape. And now, look at the intersection and watch the color squirt out the hole. Here's another work of art that deals with acuity or differences in acuity. So I wrote this book about vision in art, and my editor said that it was obvious that I knew a lot about vision, but it was equally obvious I knew nothing about art history. So he told me I had to read Gombrich, which is a really good art history book. But I didn't get past the Renaissance because I got stuck on the Mona Lisa. Gombrich says that you should look at this, that there is a reason why this is such a beloved painting. You can barely get near it at the Louvre, but it's worth it, okay? So this effect is even better in the original, but it's still pretty good in just about any reproduction you can find. Gombrich says she is uncannily lifelike. She seems to change as you look at her. And so I started looking at the Mona Lisa and sort of looking at the background and thinking about her expression, and then looking at her mouth and thinking about her expression. And I noticed, see if you can do this, if you look at her eyes and think about her mouth, and then look directly at her mouth, do you find that she seems to be smiling more when you're looking at her eyes than when you're looking at her mouth? Look back and forth between her eyes and her mouth and see if her expression doesn't change as you do this. So Gombrich says, as do most art historians, that the reason for her changing expression has to do with the fact that Leonardo used sfumato, which means he made her mouth blurry. But that just means that her smile's ambiguous, and it's left to your imagination how much she's smiling. But you're seeing something that has to do with where you're looking. That's not your imagination. That's real low-level stuff. It turns out that your acuity falls off dramatically from your center of gaze. So if you're looking at that dot, all these letters are equally readable. So your central vision is really good at seeing little tiny detailed things, high spatial frequencies. Your peripheral vision isn't just a bad version of your central vision. Actually, your peripheral vision is better at seeing big, blurry things, low spatial frequencies. So if you filter the Mona Lisa in such a way that you see how she'd look to your peripheral vision compared to how she'd look to your central vision, if you could see the whole thing with your central vision all at once, which of course you can't, you see that she's smiling a lot more to your peripheral vision than she is to your central vision. So you move your eyes constantly. As you move your eyes around this painting, whether her mouth is in your central vision or your peripheral vision changes. So as you move your eyes, her expression changes, which gives this painting a dynamic quality. And a dynamic quality in a painting 500 years ago was very special. And I think it also gives it a coy quality. You're looking at the background and she seems to be grinning from ear to ear, and then you try to catch her smiling and she stops every time you look at her mouth. You can also see this in Pointillist works. So the Pointillists made paintings made up of all these little dots of paint. They claimed that there was a sparkly quality to the colors because of additive color mixing, but magazine illustrations are made up of little dots of color, and there's nothing special about the colors in a magazine illustration. I think the sparkly quality in Pointillist paintings comes about because your central vision sees the dots, your peripheral vision sees the river and the sculler and the trees. And so as you move your eyes around this painting, what part of it is comprised of dots and what part of it forms a scene changes. Again, a dynamic quality in a static image. And I really like this one because if you're looking at the water here, you see dots of paint. If you look over here, you see lines of reflection. And if you look even farther away, you see a solid. So that's just what water does. Sometimes it's dots, sometimes it's lines, and it changes depending on where you're looking. Chuck Close took this to an even larger scale. With your central vision, you can only resolve these tiles. With your peripheral vision, you can see Chuck Close. And photomosaics. Your central vision sees baseball cards. Your peripheral vision sees Babe Ruth. So now I want to talk a little bit about how we see objects. So I've talked a lot about like early visual processing and center surround and local stupid opponency. Now I want to talk about how do we recognize faces. So we are very good at recognizing faces. You are seeing these images in 1/10 of a second. You are not only detecting faces, you're recognizing particular individuals. And you can do this whether these images are cartoons or degraded or occluded, and you can do it better than any computer program. So again, artists are ahead of us in knowing what might be going on in terms of how we recognize faces. So I got to get a new slide. A veridical line drawing is harder to recognize than a caricature. Why do caricatures evoke an identity so effectively? They look less like the individual than a line drawing, and yet they look more like that person at some level. So both humans and monkeys have special regions in their temporal lobe dedicated to processing faces. Monkeys, like humans, use faces to recognize conspecifics and to convey important social information. So 2 postdocs in my lab recorded from neurons in these face-selective regions in monkeys. And a face cell in a monkey, first of all, in these regions, they're just chock full of cells that respond selectively to faces, but they will respond to monkey faces, they'll respond to people faces, and they will respond to cartoon faces. Anything you'd call a face, they'll respond to. So we designed this cartoon face space where there are 20 different parameters of ways you could vary these cartoons that included mouths and eyes and hair and eyebrows and arrangements and shapes and all kinds of stuff, anything we could think of. And we varied these things all simultaneously and showed them to cells and said, what do these cells want? If they want just one particular face, we never would have seen it. But if they're tuned to face parameters, face variables, then we could see it. And indeed, we found, so this is the stimulus set, just so you notice that we're getting way outside of plausible primate space, face space with these stimuli. So here are 20 graphs from one face selective cell showing all the different parameters that we varied. What we found was that each cell cared about a few parameters. They didn't care about one face. What they cared about was 2 or 3 of these variables. And they were different variables for different cells. So here's a cell that cares about 4 of the variables. It cares about eye size, the shape of the eye, and the shape of the face. But what's noteworthy is that in each case, the cell is peaking at one extreme and suppressed at the other. So here is spontaneous firing, here's excitation, here's inhibition. So these cells show ramp-shaped or extreme tuning. Now you might imagine if you were going to have a cell that cared about inner eye distance, for example, you might want to have a cell that peaks at big inner eye distance, a cell that peaks at intermediate inner eye distances, and a cell that peaks at small inner eye distances. Instead, we found extremes. That's more efficient because all you need is one cell that likes big inner eye distances and one cell that likes small ones, and you get everything in between for free from the ratio of the 2. And that's what we found. But ramp-shaped tuning is consistent with caricature because face cells tell you how a face differs from the average face. And as I said, artists figured this out. Even great artists, not just the guys that you find on the sidewalk at the fair, but somebody like Picasso. So Picasso portraits look weird, right? But here are a bunch of Picasso portraits next to photographs of the people that they're portraits of. They're all caricatures. So this is Gertrude Stein, Igor Stravinsky, Picasso himself, his art dealer, some guy he roomed with, wife of a friend, girlfriend, mother-in-law. In each case, he has extracted what is unique about each of these individuals. So, ramp-shaped tuning also means that face processing involves within-category comparisons. That is, you are comparing the face you're looking at to some internal standard of the average face. That means that face processing, like everything else I've told you about tonight, is local and opponent. What do I mean by it's local? It's in faces. Well, in early visual cortex, visual space is mapped across the cortex. But when you go to higher levels in the temporal lobe, you have regions that are selectively devoted to particular object categories. So you could have an area that cares about faces, no matter where they are in the visual field, or a part of the temporal lobe that cares about body parts. So locally, in higher levels in the visual cortex, means within category. So you have within category local comparisons. And that also means, now while I'm talking, I want you to look at these numbers, okay? And the reason you're looking at these numbers is because I want to give you an after effect in face processing that's similar to the after effects you get in color and motion. And you're moving your eyes because I don't want you to get some stupid little retinal adaptation. I want you to get a high-level identity after effect. So now I'm going to show you two identical morphs. And I hope you got an identity after effect. So again, vision is information processing, not image transmission. Artists have figured out what these stages, some of these stages in information processing, and take advantage of it. You can even see this information processing in the art of kids. You ask a 3-year-old child to draw you a person. You will get a face on legs, right? Any 3-year-old child will give you a face on legs. This is a drawing a child made of his pregnant mother. A face on legs inside of a face on legs. These kids are telling you what's important. What is the information they have taken in about the identity of other people? What's important? Their faces. So that's what they tell you. I get invited to some peculiar meetings, and one of the peculiar meetings that I got invited to was a bunch of anthropologists who are carrying on about this 35,000-year-old Venus figure or fertility figure, and they were talking about all of its mystical, symbolic significance, and I'm sitting there thinking, you ask a guy, any guy, what are the important... Matisse recognized this computation. He says, I don't paint things, I paint the differences between things. I got invited to another weird meeting at the Museum of Art and Design in Manhattan, and it was a meeting about dioramas. And I almost didn't go because I thought, my kids are out of middle school, I don't have to make dioramas anymore. But I went, and I was really glad I went, because dioramas are fascinating. They're these scenes of stuff that is ordinary or even ugly, like urban decay, and yet they're exquisite. And I had to think, well, why are these things so interesting? What's so special about them? And I started thinking about the way our temporal lobe is organized into these special domains. And we know our temporal lobe is organized into these special domains because of the kinds of agnosias people can get. People with strokes or wounds can lose the ability to selectively recognize small manipulable objects or big things. Places is very common. Faces is very common. Text, you can lose the ability to read by a lesion in a particular part of your temporal lobe. That means that there are regions in your temporal lobe dedicated to processing these particular object categories. And some people think that these regions are innate, that they're just for things that are biologically important. But some of the research in my lab that's going on now suggests that there may be a huge experience-dependent component to this. That is, our brains are designed to learn to be good at recognizing things that we are exposed to. And I think that by having clusters of cells dedicated to processing the same object category means that you have cells next to each other that can inhibit each other, which means that you have fine within category comparisons. So if you have these special areas, and I want to convince you that you have one of these special areas and that it makes you an expert at recognizing faces. Now, you've seen this illusion. This is the Margaret Thatcher illusion, was invented by Peter Thompson. And you know that I've done something to the one on the left, right? But it's okay, right? It's nothing really weird about it. But if I turn it, now you're processing it with your face cortex. Now, you saw that very differently when you were processing it with not face cortex, because it didn't count as a face. It didn't get into that module. It didn't get into your special module because it wasn't an upright face. But now it's an upright face, and you're processing it like an expert, and you're processing it differently. And I think that artists have figured this out and have figured out ways to get us to look at things with different parts of our brain. They're always saying that they're doing this, you know, that they're trying to get you to look at things differently. Literally, they are. They're getting you to look at things with different parts of your brain by making it the wrong scale. That's a big one. Or just putting it in the wrong place. Now, remember how hideous that looked. Does it look that hideous anymore? No. Isn't that weird? It kills me every time I see this. So another thing that I think is interesting that artists may be way ahead of us neuroscientists is the fact that the world looks stable, right? You look out on the world, it looks perfectly stable. I know it is stable except for the, moon and stuff, but you do this with retinal images that are constantly moving, right? You move your eyes 3 times a second, 2 or 3 times a second. So your retinal images are constantly changing. How do you knit this together into a constant perception? So I want you to close one eye and just tap, wait a minute, close one eye and tap on the open eye, the lid of the open eye, and you'll see the world jump. That's what your retina is doing constantly, moving all the time. Here you can see your eye movements. I don't know why this works, but you can see your little eye movements a few times a second in the way this thing shimmers. Don't ask me why it works, it just does. If you don't move your eyes, things fade. So there is a purple spot up here, but if you fixate on this cross for just a few seconds, the purple spot goes away, and if you move your eyes, the purple spot comes back, right? That's fading because the edges are blurry. So if you don't move your eyes, or if you stabilize the image on your retina, you stop seeing things. So in the same way that center surround cells make you sensitive to discontinuities in space, they also make you insensitive to things that don't change over time. So that's the reason why you move your eyes around, is so you can refresh the image. Here's some fading. If you look at the top P letter, I hope in just a few seconds the whole thing fades to pink. And now look at the bottom, and now you can get the whole thing to fade to blue. I think Rothko does this. His paintings are designed to be viewed in rooms that are very small, so that you have to have them fill your whole visual field, and the edges are invariably blurry. And so I think you get this, what's called Troxler fading, when you look at a Rothko. And there are these 2 paintings out in the hall by Janet Rosner that I think do the same thing, that are fun to look at. But let's go back to how do we perceive a stable world when our retinal images are constantly changing? Again, I think that artists are ahead of us neuroscientists, and the artists who are ahead of us right now are the people who do film editing. Because when you're making a movie, you have the same problem of moving a camera and cutting from one scene to the next. And a rule, the most important rule in cutting, is whenever possible, cut in movement. And that's because when you move your eyes, you don't notice when things change. So here's an illusion by Peter Tse that I hope will demonstrate this. Those spots are changing, right? But if you track that little red dot at the bottom, I hope you notice that as you're tracking the dot, the movement of the big circles is attenuated. Do you see that? So as you move so that may be part of the answer. You move your eyes, you ignore changes, because the changes are just due to retinal movement. Here's a little clip that I found on the internet explaining how you cut in motion, but what I want you to notice is the cut at the end. It's really good.
Here are 2 shots, one wide and one close up. Cutting on motion allows you to go from a wide shot to a close-up in what will appear to be a seamless edit.
Wasn't that cool? I love that. They do it all the time. I can't watch movies anymore. I'm so boring. I say, look at that cut. So the visual system ignores changes that occur when you move your eyes. But you now have to link together what happened in one frame, one viewing, to what happened in the next one. So there are perfectly intelligent people in my field who think that you remap the visual world. You keep track of where your eyes moved, and then you rearrange the Cartesian coordinates some way. First of all, that's going to be really hard to do. And second, there's no way you could do a zoom, right? We're not designed for that. So I don't think that happens. I think what happens is that you link together things that look the same, and more importantly, you link together things that move the same. So again, you're not going around checking the laws of physics. If there's something that's going across your visual field, you don't check to make sure it's the same object the entire time. And here is a demo of cutting on motion. Now, the last thing I want to talk about is stereopsis. So for those of you who have stereopsis, just to remind you, both eyes open, blue on the right. That's stereopsis. Some of you don't have stereopsis, don't worry. But both eyes open, blue on the right. Okay, that's all. It's not that interesting. It's just geometry. If you're looking at this dot, it casts images on corresponding parts of your two retinas, but objects that are closer or farther away than your fixation plane cast images on non-corresponding parts of your two retinas, and your visual system uses the differences in the images in the two eyes as part of its computation of distance and depth. So if you go to a museum and you find a nice painting that's got lots of shading and perspective and depth cues, part of your visual system is saying, nice three-dimensional painting, and the rest and your stereo system is saying, no, it's flat, I'm sorry. Your stereo and your relative motion parts are saying it's flat. So if you go to a museum, if you want to fall into a painting, which is kind of fun, close one eye. But stereo matching is a computationally difficult problem. So if I tell you, don't use the glasses yet, that the cyan spots are white to your right eye and the red spots are white to your left eye, can you see what this shows? No, it's hard. It's a really but your visual system, again, both eyes open, blue on the right, your visual system can do this computation instantly. And some of you can't see this, don't worry, okay? So some of you saw it, some of you didn't, don't worry about it. So stereo matching, when you've got a bunch of identical images out there in the real world, you've got 4 images on each retina, and this is the correct match, but you could match up number 3 image in the right eye with number 4 image in the left eye, and it would correspond to the same object, but at a different depth plane. So if you have ever misstepped on an escalator, you did it because escalators, for some stupid reason, are made with these slats that you can misfuse in your two eyes, and that would correspond to the step being at a different distance. I think the post-impressionists and the impressionists, they were always talking about painting the air. I think they did it using these dabs of paint that can be misfused in your two eyes, and so that it can look like a volume of speckly stuff. And this thing by Klimt, I find it almost nauseating the way the leaves misfuse in my two eyes. But what's peculiar about this one is that Klimt himself had no stereopsis. Now, how do I know that? Well, look at his eyes. He's cross-eyed. You can't have stereopsis if your eyes are crossed or misaligned because, as I said, the computations for depth begin in early visual cortex where they are mapped retinotopically, that is, across the visual field. And if your two eyes are pointing in 2 different directions, the information from the 2 eyes about the same part of the visual field don't even end up in the same part of the cortex. So you don't have any depth perception. I got interested in this issue when I did some research on people with dyslexia, because I found that equiluminant writing shimmers and is difficult to read, and I remembered that people with dyslexia, which is a selective problem in learning to read, there are a number of people with normal intelligence, normal education, who just have a terrible time learning to read. And they complain that normal text, black and white text, shimmers and moves on the page. And I thought, well, whoa, something about maybe their wear system might be odd. And so Al Galiberta and I measured the timing in the visual systems of some people with dyslexia, and we found a slight slowing in the wear system in people with dyslexia, which may have to do with moving your eyes and remapping the scene or stabilizing the world or something like that. But whatever, remember, what are the functions of this system? Motion, depth, spatial organization, and figure-ground segregation. Well, what are the symptoms of dyslexia besides problems learning to read? positional information, figure ground segregation, and trouble gauging distance and depth. There are also auditory issues, and this may account for the phonetic problem that people with dyslexia often suffer from. So differences in ba and da in particular happen really fast, like 40 milliseconds. And so there may be a fast and a slow subdivision in your auditory system as well, and dyslexia may affect the fast subdivision in the auditory system, which would give you auditory problems in phonetics. But when I did this research, I was privileged to meet a large number of people with dyslexia. And it was inescapable that many of these people were seriously talented. There were a number of artists, musicians, actors, and computer programmers. And it's been documented that dyslexics are overrepresented in the artistic population, but the usual explanation is that they were no good at academic subjects, so they spent all their time in the art room. But I had to think about, well, because I don't care how hard I worked at it, I couldn't have been as talented as many of these people. So I thought about, well, what might make them unusually good? Maybe there's something about the dyslexic brain that makes them bad at reading but good at music and good at drawing. So the auditory thing is easy. Phonetics is fast, music is slow. Maybe they are just better at slow transitions. What about drawing? Why are so many artists dyslexic? Well, maybe trouble gauging distance and depth would be an asset for an artist. I can't draw a chair because my brain has processed the information and made it three-dimensional, and I can't get back to the flat retinal images. Well, maybe if you see the world as a little more flat, you're better at seeing monocular depth cues and flattening the world onto a canvas. So we looked at a bunch of artists. Here's Chuck Close, severely dyslexic, and probably has terrible depth perception because his eyes are different magnifications. You can tell if someone could have normal stereopsis just by looking at a photograph that's got a good reflection of light in the two eyes, because if the light reflection is in the same place in the two eyes, their eyes are lined up, and they could have normal stereopsis. So here's some baseball players, right? You see the light reflections in exactly the same place in their two eyes, right? Are you good at this? Now, there's one baseball player who had no stereopsis. Light reflexes centered in this eye and off to the side in this eye. Babe Ruth had amblyopia. So if when you're little, the input from the two eyes doesn't end up in the same place, you've got a choice. Your brain has a choice. It can either alternate which eye it looks through, those are alternating strabismics, or it can ignore the input from one eye, the connections from those eyes wither away, and you have what's called amblyopia, and you have very poor vision in that eye. That's what Babe Ruth had. So we got photographs of several hundred famous artists and looked at their eyes. Now, I told you that 10% of otherwise normal people have no stereopsis, but only 3% of the population has significant strabismus. You would expect that we would have found maybe 2 or 3 people, artists, who didn't have aligned eyes. Instead, we found dozens. Here's Andrew Wyeth. This eye's looking at you, this eye's looking over there. His father, N.C. Wyeth, also an illustrator, also wall-eyed. Edward Hopper, light reflex next to the pupil in this eye, on the edge on this eye. Carol Walker, every picture I've ever found of her, the light reflex is in different places in her two eyes. She works in silhouettes. How flat can you get? I don't know about the dog. Mark Chagall was cross-eyed. Frank Stella was cross-eyed. Jasper Johns was wall-eyed. Robert Rauschenberg was wall-eyed and severely dyslexic. Alexander Calder was wall-eyed. Man Ray was wall-eyed. And Frank Lloyd Wright was wall-eyed. And then there are a whole bunch of others that you haven't heard of, but are also strabismic. So as I said, there's an actual clinical test, the Hirschberg test, where you're allowed to look at a photograph and diagnose strabismus. You're obviously not allowed to do this from a painting because, you paint the eyes at different times, and I told you your eyes move 2 or 3 times a second. But I was at the Louvre, and there's this little room they have there that's just got 4 Rembrandt self-portraits in it, one on each wall. all 4 of them had one eye looking straight ahead and the other eye looking off to the side. Now, I know when you paint a self-portrait, you look in a mirror. So here's a montage of me. I'm looking, and I'm holding a mirror up about 8 inches from my eyes, and here I'm looking at my right eye, here I'm looking at my left eye. And I have normal stereopsis, but the light reflex is in 2 different places in my 2 eyes. But my eyes are nowhere near as deviated as Rembrandt portrays himself. So I had this pile of pictures of Rembrandt self-portraits in my office, and my young colleague, Bevel Conway, who is himself a stereo-blind artist, he's an alternator, said, "Is it always the same eye?" And I said, "No, if it was, I could publish it." Sometimes it's the eye on the right side of the painting, and sometimes it's the eye on this side. And Bevel, being a stereo-blind artist, said, "Marge, you have to separate the paintings from the etchings." So when we did, we discovered that in the paintings, it's always the eye on this side that deviates, and in the etchings, it's always the eye on this side that deviates. When you make an etching, you scratch it on a plate, and then you flip it over, okay? So we made a graph. We used this little program where we found the outline of the eye, and then we outlined the iris, and then we figured out where they were relative to each other, and we made a graph. If you can make a graph of the unlikeliest thing, you can get it published. So in the paintings, it was generally the eye on the right that was looking straight ahead and the eye on the left that deviated, except for this painting, which is in Berlin. Maybe it's a fake, maybe it was made from an etching. And in the etchings, it was generally the reverse. So that suggesting, confirming our idea that great artists that stereo blindness may not only not be a disability, it may even be an asset for an artist. Now, you do not need to go poke your kid's eye out if you want him to be an artist. The first thing you learn in drawing class, you want to flatten the world, close one eye. But maybe in little kids, they don't go around telling them to close one eye, and the kids who see the world as flat have a little advantage over their peers and just get that much of a head start in trying to become an artist. But in any case, so here's some take-home messages. You don't really need these take-home messages, except that people are different, and something that may be a handicap in one task may be an asset in another. I think that's kind of important for people to remember. And if you're not too attached to them by now, could you please leave the stereo glasses on the table outside on your way out? And I would be happy to take questions.